For my mini project for SOLUR, I decided to highlight two new lab techniques that I learned during Immersion Week back in October. I picked two of my favorite that I learned throughout the week of immersion. And our little overview for today is, like I said, I'm going to talk about my two lab techniques that I learned about during immersion. And during this presentation, I'm also going to show you a little group video of me talking about one of my two favorite lab techniques that I learned about. So the first one that I'm going to talk about is cell passaging and cell seeding. I found this one very interesting, especially since it's one of the most important steps that you can do in a lab when it's concerning cells. So the importance of cell passaging is that it does maintain and keep the cells healthy. It also maintains their exponential growth, and it helps continue to promote proliferation within the culture. So it helps make sure that the cells are still living and that they are continuing to grow while you incubate them. To prepare for this procedure, one of the things that we had to do was note the level of confluency of the cells and the date in which the cells were last fed. That is very important because we want to make sure that our cells are continuing to thrive. In order to prepare for that, you want to make sure that you have the proper concentration of trypsin from a frozen stock that we pulled out of the freezer in immersion. And then you want to make sure that everything is temperature equilibrated and it's all the same temperature so that you are not killing these cells. You also want to make sure that you have a proper level of serum. You want to have the correct antibiotics ready. Everything needs to be sterile. That is one thing that they emphasized with us, is that making sure all test tubes are sterile, the plates are sterile, you have sterile plastic, and you have sterile glass pipettes. And then they also want to make sure that you know how to clean the hood based on aseptic cleaning protocols that we learned. That was the first thing that we learned. And I actually found that pretty interesting as well, too, because without that, then you're disrupting a very sterile environment, and that can actually disrupt the cell passaging and seeding that we are about to perform. So on my next slide, there will be a video of me kind of talking about this little slide right here. So let's go ahead and check that out. Let me go ahead and play this, and then I'll talk about the procedure that we did for this technique. So unfortunately, it was only 10 seconds long, but that was me just talking about cell passaging. So once you get everything clean and you can do the aseptic techniques, the first thing you want to do is you want to aspire out the old PBS media that was in there. That actually helps keep the cells moist and alive. In a dry environment, they won't survive. And then once again, you'll wash them with another round of PBS just to make sure that, you know, they're still staying moist, get everything that was out from the old medium out there. And then you'll aspirate that again, which means just to suck it up through a little vacuum that you use with your foot, which is actually pretty cool. And then you will take that and you will add 1 milliliter of trypsin into the cells, and you're going to leave that in there for 5 minutes. So what we did is we basically just rocked the cells back and forth in the hood, which was actually just moving them so that you can mix the trypsin all around. And then once that is done, you just want to keep it going. And then what you're going to do is after that is you're going to aspirate the media again, and then you're going to add 2 milliliters of DMEM, which is actually going to inactivate the trypsin that you just put in, and make sure that, you know, there's no active trypsin into those cells. And then once you do that, you're going to centrifuge your cells in for 5 minutes, just to kind of mix everything up, get everything good and mixed and all that. And the one thing that you want to make sure is that when you're using the centrifuge, we were emphasized that you want to make sure that it's even. So if you only have your tube, you want to make sure that you have another centrifuge tube that is the same weight, so it balances out the centrifuge. If not, it's not going to centrifuge correctly and it's going to be all wobbly and everything. So it was really cool to see how that worked as well. And then once that's done, you're going to aspirate the medium that was in there, because what it's doing is it's separating the liquid from the cells, and the cells are actually going to be at the very bottom of the centrifuge tube. And you're going to aspirate that media so that way you only leave a little pellet. And it was really cool to see that little pellet underneath to see that, oh, these are all the cells that we just took out from the plate from the PBS media and the trypsin. And then you're gonna add some more PBS into there once you add that little pellet in there. So you're gonna add some more PBS into the centrifuge. And what you're gonna do, what we did is you actually took the pipette and you kind of piped it up and down to resuspend the cells in the new PBS liquid, which seeing that done in real time was actually pretty cool because you're actually seeing the pellet like dissolve and the cells being remixed in. And then what you'll do is you'll pipette that into another plate and you'll be able to actually view your cells on a computer screen to see them inside the plate. And I think the cells that we looked at were fairly, they were all pretty much just on the plate. And it was really cool to see cells in person with using cell passaging. And one thing that I really liked is how simple this, well, not simple, but how straightforward this technique was. It actually taught me that doing cell seeding is very important in the lab. And it was just, I wanted to keep doing it. So the second technique that I'm going to describe to you is P-glow bacterial transformation. When I saw the sheet for this one, I actually got really excited because I kind of wanted to always do an experiment like this. So what we did is to describe the experiment a little bit, P-glow is a plasmid that is used to look at genes of interest that express GFP. And in order to see this transformation, we introduced this P-glow and the GFP into E. coli. And then the P-glow that we had was either a negative P-glow or a positive P-glow. So one of them didn't have P-glow in it, one of them did. And they were mixed with the E. coli. So that way when we put them on the plates, we were going to be able to see any bacterial colonies once we let them incubate. So we had 5 different plates for this one, and we had to label them positive P-glow LB, positive P-glow LB amp, positive P-glow LB amp aura, negative P-glow LB, and negative P-glow LB amp. So for the LB ones, that is just the medium that goes on the plate. So that didn't have any of the extra add-ons like the amp or the amp aura. AMP is short for ampicillin, which is a enzyme that selects the bacteria and it has the plasmid in it to help see the GFP, to help see the bacterial colonies that we're going to be looking for this experiment. And then the last one is ARA, which is short for arabinose, which is a sugar that helps amplify the GFP that we will want to see when we put this on a black light or underneath as you can see down here, we're going to want to see the glowing for it when we actually look at the bacterial colonies. So once we did that, they were spread across the plates. And the way you actually had to do it was you took a little rod, you dipped it into either the P-glow, the negative P-glow or the positive P-glow, and then you took each little plate and you just went back and forth like this, horizontally, and then you went vertically, and you wanted to make sure to do it until it wasn't super shiny anymore, which means that you got it all over the medium inside the plate. And then what we did is we put all of them together, we stacked them, and we put them into an incubator for 48 hours to see if we would have any colonies produce. So we expected to see growth on the plates that had the positive P-glow with the LB and the AMP, and the LB and the AMP aura. So as you can see in this picture right here, which is really, really cool to see, like everybody's was different. You can see that the LB amp aura has the most bacterial colonies because you have the ampicillin, plus you have the sugar that's also helping amplify these bacterial colonies. And then you can see the LB amp has some colonies, but not as many as the LB amp aura. And then you have the LB, the positive LB with the positive P-glow, and then the 2 negatives where you don't really have any bacterial colonies, which was kind of expected just because we didn't have the extra 2 components as we did with those 3. So we really didn't expect any bacterial growth. And then what we did too was that we surveyed everybody's bacterial cell growth within their plates to see how it was distributed through the class, which the results were actually pretty interesting to see. So as you can see, on average, we had about 5 bacterial colonies with the LB amp aura and the LB amp. Or with the LB amp aura, as you can see, roughly about 5 colonies per plate. And then for group 4, group 2 had about 4.5. And then you can see with the LB amp, there wasn't as many. So definitely having the arabinose in there helped increase the bacterial colonization or colonies that we saw. And then we wanted to see the efficiencies between, like, we wanted to see like how well everybody did within getting their bacteria to grow. And as you can see, we had almost about 200 for the LB amp aura between group 4 and group 2. And then we had pretty much in between 100 to 150 for the LB amp, which is kind of not surprising because that arabinose really does help bring out more of the bacterial colonies. So I really enjoyed doing this one because it was very interesting to see how bacteria develops within like certain parameters, such as having the piglo or having the ampicillin and the arabinose in them. I would definitely love to do more of this technique in the future, just because it was one of my favorite things to do. Is really interesting to see how this is done, and see the total procedure go full out for the 48 hours and then come back to it and see that we had bacterial growth. Other than that, this is my little mini project of the 2 techniques that I learned while being at Immersion and through SOLUR this session. I've definitely enjoyed my time in SOLUR, and I cannot wait to continue on for the next session. So thank you for watching my little presentation about my 2 favorite techniques, and I hope you all have a great rest of the semester.